All right, so today in lecture 15, we pick up basically where we left off last class, talking about Markov models and hidden Markov models for gene finding. Before we start with that though, I'd like to cover a few little things about R, in particular related to iteration. And then, like I said, we'll go to the my notes for hidden Markov models, and then we'll come back to some slides that look at other aspects of gene finding that are used in practice and some of the, sort of industrial strength software that's out there. Okay, so iteration, which, we've already seen a few examples of it in different formats, different forms, including a few for loops and some apply statements here and there. I wanted to cover it a little bit more carefully here. Okay, so let's start with for loops. They're particularly useful if we know a priori, so in advance, how many times we need to execute some body of code or something, right? So a body of code in R is anything surrounded by two curly braces. In computer science, they really like their braces and they name everything. So you have parentheses, okay, I won't write that out, but that's a parenthesis. And we know that that's very different than a brace or a square bracket. And so we might call that a bracket, or explicitly a square bracket. I would say, and I won't write it all out because my typing's terrible here. And then a curly brace. Sometimes we just say curly brace or curly bracket, right? And they have three different meanings. We know that the parentheses are used for function calls, for example, or for forcing the order of operations. Brackets are used for selecting. We've seen both the single bracket and the double bracket for selecting elements of a list, for example, or columns of a table. And the curly braces are used to denote a body of code. So all the code, in this case here, just one function call to the cat function, belongs to this same body that from the opening to the closing brace. And the for loop here has the following structure. It's ubiquitous. We have a for statement. It's not a function. It's a bit different than a function, so to be a bit careful. And then there's a space here, but then this says that for I in 1 to 10. So we know what this means, right? That this guy here, we've seen this many times now in the course. This is just then a vector 1, 2, 3, ... 10, right? Okay, so I is going to take the value 1, and then it's going to take the value 2, and then 3, then 4, then 5, all the way down to 10, okay? And it does this in this order. So basically this specifies a range of values for the variable called I. All right, and this I is like any other variable, essentially. It takes, in this case here, if we go back to our canonical model of what happens in R, it creates a box, and at the beginning it puts 1 in there, and then it's going to put 2, then 3, etc. So how it does that? So it comes in here, and firstly I will set be equal to 1, okay? And now it'll execute this body of code, which is just the function cat to send to the screen. Good job, Aki, you've lifted it 1 time, okay? Oh, sorry, this is in response to the question, how many reps of a 20 kilogram weight can Aki bench press? And so we're going to count them through. So he's lifted it 1 time because I is equal to 1. And then it comes to this closing parenthesis, and now it goes back to the opening parenthesis. But it updates I, and so now I will be equal to 2. And now it executes this body again. In this case here, I is going to be equal to 2. And so it prints out 2 times, and it hits the bottom, goes back to the top, prints out 3 times, all the way down to the last value for I, which is 10. It comes into the body of code, executes that, and then it stops. And then this piece of code down here will execute after this for loop has finished, okay? So we knew a priori that Aki wouldn't be able to bench press the weight more than 10 times. And so we could count with him 1, 2, 3, 4, all the way to 10. Okay. Another example. Okay, so sorry, in general, for loops have this structure. Whoops, and I missed a parenthesis there. For, and then the variable name in our last example, that was I, in, and then a set of objects. In the previous example, we saw that the set of object was the integers 1 to 10. But it could be anything. It could be names or logicals. It could be basically any object that we would like. And that's what this variable I, or in our case, that example was I, whatever our variable name will be, will take on that set of objects in that order. And each time it'll execute this body from the beginning to the end and redo that. Okay, so yeah, I guess like traditionally for loops in R were discouraged because it slowed things down, just the nature of the language. But that's been fixed for a while now, and there's absolutely nothing wrong with for loops, and I find them that they're very intuitive, but other people don't. So some people get a little bit confused by them. So here's a second example. Now I've set a vector of characters, character vector, right, of all boy bands that I could think of, and then for. Here's our for structure, and this is our body, open, and our close is down here. And inside of it, we actually only have one statement, which is an if. Okay, so in this case here, if you detect in the bth element of bands the word Justin, it executes this statement. Otherwise, it executes this statement. So this is an if else statement, which we've seen last class, right? So now we're making a conditional. If the boy band starts with, it has the string Justin in it, it will do one thing. If it doesn't, it does something else. But let's take a look back here. Bands is a character vector. So if I want to access, like, say, the third boy band, it would be this guy here, and this would be the third element of bands, right? So now what's happening here is that B ranges from 1 to the length of bands. So the length is going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So B is going to start at 1, and it's going to go up to the length of bands, which is 14. So here the string detect is on the bth element of bands. So the beginning, that's the first. And so the first guy is Jackson 5. And when it comes in here, the string detect will be, let me clean this up a little bit, okay? The string detect will come in, and it'll fail because Jackson 5 does not contain Justin, and so it'll come down here to the else statement and execute this. So now we print out on a scale from 1 to 100, Aki rates the bth band, which is going to be the Jackson 5, so that's the output right here. And how much does he like that between a scale of 1 and 100? Well, we use the random function, the random number function. So I choose a random number between, 95 and 100 because he loves all these bands. And that's the sample function. And you can see here that Aki loves Jackson 5, or rates them as a 98 out of 100. Then we go back to the beginning of the loop. Maybe I have to be in, oh, I see. Okay, I got the hang now. It just took 16 lectures or so to figure out this pizza thing, but I got it. So now it goes back to the beginning, which is up here, and now B becomes the second element in bands, so that's going to be, scrolled off here, New Kids on the Block. It doesn't contain Justin, so now it comes in and executes this guy down here again. And again, now it selects a random number, and it turns out that Aki rates New Kids on the Block a 96 out of 100, so not quite as good as the Jackson 5. And then it goes back to the top. Now eventually it will get to number 1, 2, 3, number 4, B will be equal to 4 down here, okay? And the string detect actually determines that the fourth band does contain Justin, and so it executes this, and Aki rates Justin Bieber to be infinite. Okay, infinite like, maybe love. And it will go through the rest of the list exactly like that and execute each one. And so we'll see something, all of Aki's favorite boy bands down here like that. Okay. Okay. The way I had done this from 1 to length in the previous, I set the range of B to be 1 to the length of bands. And that can be a little bit dangerous. It's better to use this function called seek along, okay, and it's a little bit safer. Okay, so well, this is, you can ignore this. It doesn't actually work in my slides. It should work in your session. If you want to change your output text different colors, you can use this crayon package. It's really not really important here. So here. Aki's pumpkin carving success is going to be the empty set, right? And you can witness that down below with this picture. And now, but the thing is, if I ask for, let's suppose I wanted to go and create a for loop, and I wanted to say every time that Aki successfully carved a pumpkin with some skill, I'd say, wow, Aki, that's really interesting. But he doesn't have any successes, so this shouldn't print out anything. However, if I use this 1, if I use P to be 1 to the length of pumpkin carving successes, I get some strange behavior, because 1, that's this guy here, go to 0 is the length of pumpkin carving successes, because he clearly has no successful pumpkins carved. But still, when I run this, it executes this twice. It first prints out, wow, Aki, that's really interesting, and again, so P takes on the value 1, and then it takes on the value 0, which isn't really what we wanted, right? The thing is that you can count down. So R tries its best to understand that. It says, oh, you're counting down from 1 to 0. That's not what we wanted. We wanted to basically say, if there are any successful examples of Aki successfully carving a pumpkin, then we'll go through the for loop. But since there's not, we don't want it. Now, I can rectify that situation by using seek along. Here, again, Aki has no skill carving pumpkins. If I ask for seek along, I get back now just integer 0. So if I say for P in seek along of pumpkin carving successes, now there's nothing there, and there is no output at all. And this makes a lot more sense. It corresponds to our notion of reality, right? Okay. Now, in general, there are other ways, several other ways to implement iteration in R. The for loops are part of the base package, and we'll come back and I'll show you while loops in a little bit. They're also part of the base package. But, well, per is a package within the tidyverse. It has lots of nice functions for iteration that make things a lot easier. Here, I'm just loading the Tara data set, our Tara Oceans data set, and I'm going to select all the columns that begin with bacteria. So that's all the species, all those columns to the past salinity, oxygen, etc. Okay, so that's just a straightforward select. I just use the starts with function to select those columns that are bacteria, and then I'll pump my Tara small, Tara S, into this function called map. In this case here, it's map double. Double is basically a synonym for numeric. There's different double functions, one for logical, one for double, one for characters, etc. Here, if you know your data is a double or a numeric, it's the right one to use. And so what it does, when you pass it, when you pipe this tibble into map double, is it sums every column. Okay, which you've seen different ways of doing that now, but here I can sum each column, and I applied the sum. Now here, sum takes this other parameter called NA remove, and you need that to be true because there's lots of NAs in this data. That's map double basically will pass this part on to the sum function. It's like it's called you'll hear this term sometimes called the triple dot, and that means basically whatever else you pass in the map double gets passed as arguments to sum. Okay, and then I took the output of that, which is going to be the column sum for every bacteria that we've detected, unnamed. You can forget about this. Remember, the names are very long. They're like this bacteria, proteobacteria, alpha proteobacteria, etc. It was kind of ugly, so I put the unnamed in there to get rid of the names on those values. And now this is the first taxon that we identified. That's the total count across all different sites. So that was 102,000 or so. And I printed, that's what this is here, the printout of all the different taxa that were identified. And then I pass it to the summary function, which is kind of a nice little function. I'll scroll down here to the end. It just basically gives back some simple statistics, summary statistics about those counts, saying that the average taxon was had a count of 362. The biggest one, as we know, was 102,000, and there's many with just a single count over all sites, just detected once. And this is your first quartile and your third quartile, right? Think of that as being, the mean is the second quartile, right in the middle. The first and third are between the min and the mean, etc. And you actually see here that there's a big difference between the mean and the median. Most taxa have very few reads that identify them. Okay, that's neither here nor there. The main thing here, I just want to go back to make sure you understand that the map function, I could have written a for loop, and that would be an excellent exam question to ask you, can you make a for loop, use a for loop here instead of the map double, and it would be pretty easy to do. I'll leave that to you. It's a good take-home exercise. Maybe you want to make a note of that right now. Okay, try it out. But that map function basically says, okay, for each column in the tibble, use the sum. Here's another example using a map logical. Here, I've just done something. It's quite simple, but I pipe in my tara small into the map logical function. And this kind of strange notation, this formula notation again. Just think of it as a function, okay? And so my function here is that I sum up each column of tara. That's the notation .x. Map logical takes data, that's .x, and the formula or function, that's .f. Here I'm explicitly saying pass tara s into this sum and with the NA remove, and it'll return true if the number of counts is greater than 50,000. Otherwise it returns false. And so here you can see that it's only the first 5 taxa that have more than 50,000 reads. In fact, I didn't even know this, but they had sorted the data by, the columns are sorted by most to least observed. So you see these guys here are the ones above 50,000, and then it drops off below 50,000. And this is sorted. You can check that out. Okay, so that's the map logical function, and it works, again, very, very similar to the map double. And instead of a for loop that goes over each column, you could just, you don't need to specify where you begin and where you end. So you can see, like, perhaps in this example it's a bit clearer. You don't need to know how many columns there is. You don't need this variable like I in 1 to n call of tara s. It's all taken care of for you. It just figures out how many columns there are and iterates over them, each one. All right, and then the last concept that I want to talk about today for this little R session is the idea of iteration over of an undetermined length. And so sometimes we don't know a priori how many times we need to loop over a body of code. So sometimes it's just hard to predict how many times you're going to need to do it, So in this example, this is just a simple example. We start with a string, a nucleic acid sequence, okay, like that. And I'm going to trim from the 5' end until I get total GC content of over 80%, at least 80% at least, okay? So here I've written a small little function. This function takes a string, an S, and it returns the string count, so the number of times that G or C appear in S. So remember, this is a regular expression that we talked about a little bit here. So this means either it's a G or a C, okay? And in the string S, okay, that's passed to the function. And I'll divide that by, it's funny, I didn't think my thing was on. Okay, I'll divide that by the entire length of the string times 100. So that's the percent GC, because you know the string count is, this is the number of G's and C's, and this is the total, including A's and T's, and then I multiply that by 100. So that's just the percentage. And now I write the while function like this. Okay, I have this special command while, and then I have, now this is a condition. It's a logical expression. Okay, that's a logical expression, and that logical expression, of course, evaluates to either true or it evaluates to false, like all logical expressions do. And so here, if this evaluates to true, okay, then I execute my body of code that's determined by this opening and closing curly brace. If the logical expression is false, I stop. So here, in other words, I'm saying, as I modify the string by cutting off nucleotides from the X prime one by one, as soon as the resultant, the resultant GC content is at least 80%, then I'm going to quit. Okay, so here you can see this is the initial string. It starts off with ACTTAGCA, and it comes in and it says, okay, well, calculate the percent GC of all of X. I don't know what that is, but you can see that it's not less than 80%, because here it cut off the first, that string sub, it only takes from 2 to the length of X, so it cuts off this first nucleotide, and it prints out the resultant string. So now it's just CTT, right? CTT. Now it goes back to the beginning, okay, and it checks this condition again. And obviously it's not below 80, above 80%, because it comes in and cuts off this C, all right? And now we're at TTT, right? And it's going to keep going back, and it's going to iterate through this loop, this body of code, each time removing one character, until eventually I get down to here at the very end, where you can see that there are just 7 nucleotides, one of which is A, all right? And it now, well, now you know that 7 out of 8 is greater than 80%, so we stop, right? So there, I had no way to knowing really beforehand how many nucleotides I'd have to cleave off before I could stop. And so that's why I used here the while loop. Okay, so that's my short note on iteration for today, and a little bit of new stuff for R. Now we're going to go over to some notes for the hidden Markov models, and then we're going to come back to some slides about gene finding approaches in software. A hidden Markov model builds on top of a Markov model. Okay, so this is your typical Markov model. Nothing's changed. I didn't write the transition matrix here, but it's arbitrary. This is your nodes and your transition probabilities, and the starting probabilities for the different states. But now a hidden Markov model is a Markov model augmented with emission probabilities at each node, at each state. For example, if I'm at state G, you can think of it as like I'm emitting, or I'm writing down on paper a symbol, and that symbol will be n with probability 0.7, and and the symbol will be d with probability 0.3. These two values have to sum to 1. So it can be any number of symbols, but they would have to sum to 1. And at state A, I emit an n with probability 0.8 and a d with probability 0.2. So they don't have to be the same probabilities, of course. but each node has emission probabilities for different symbols. So that's seeming like a pretty small modification to Markov models. Why does it matter? How does this work? So let's start again. Step 0 is just like before. I would pick a random number. Let's suppose my random number would be like 0.56. Just an example. Now I might say, okay, well, because it's above 0.5 and below 0.75, I start in state G, okay? So I start in state G, and now what I do is before I transit to the next state, like I would in a normal Markov model, now I roll the die again, and with probability 0.7, I emit an N, and with probability 0.3, I emit a D. So we can keep track of this now as follows. We have two things. We have the state that we visit, and we have the symbols that are emitted. So my initial state is G, okay? And maybe I roll the die and I emit, maybe I roll a 0.5 and I would emit an N. Now, just like with the Markov model in step 1, I roll the die and I decide, okay, so this step 1/2 is the emission step. And now I roll the die, and this step here is the move step. So maybe I roll the dice, and my probability the dice rolling might say I should switch over to state C. So now this is a C. And now I roll the dice, and with 50-50, I emit either an N or a D. Let's suppose I emitted a D. That's this step here, emission, right? And so then I go back to step 1, right? And now I pick another random number that controls this is again the move step, right? So I might move, maybe I stay in state C. And now step 1.5 is I pick another number randomly to determine which symbol I emit. Maybe I again emit a D. And I go back to step 1, and now I roll the die for the move. So maybe this time I move to state A. Okay, and then I roll the dice, and with 80% probability, I emit an N, and 20% probability, I emit a D. So maybe it would be an N. Okay, so now in the hidden Markov model, there are two things. In the hidden Markov model, there is the state sequence, right, and the symbols that are emitted each step. And typically when we're modeling and using hidden Markov models, what the situation is that the underlying state sequence is hidden. We don't see that. That's the underlying steps that the person makes in this little Markov model. What we actually get to see, the public part, is the emissions, the symbols that are emitted from the system. That says, maybe that's my English. My writing isn't very good there, so that's public. Okay, so everybody can see it. Okay, so here's a challenge to understand why hidden Markov models can be used, right, or how they can be used and why they're important. So imagine you're at a casino, okay, and the dealer has 2 coins, 2-sided coins, Michelle, not like 4, 8, 10, etc. So then the dealer throws 1 coin at a time, okay? But you don't know which one. On the surface, the 2 coins are indistinguishable, but actually 1 coin is fair. It's 50-50 odds it lands heads or tails. The other coin is biased, and it lands 90% of the time as tails, okay? So we don't know which coin the dealer is using, but we get to observe whether the coin was tossed was heads or tails. Okay, then the dealer repeats the following 100 times. She puts the 2 coins behind her back, and she secretly chooses either the fair or the biased coin. Then she flips the coin in front of you and adds the outcome to a long series of outcomes, right, for the 100 trials. For example, after 1 game, you would see, well, this is really ... It should be length 100, but it would start off with heads, then tails, then heads, then tails, 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 heads, 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 tails, etc. Okay? Now, you win this game if you can predict or guess correctly which coin she used at each step. Okay? So if you saw just the heads here, which coin might you think she used? I would probably say the fair coin, because the probability that she produced the heads with the fair coin is 50%, whereas the probability with the bias coin is just 10%, right? So here she's, and then you see a tails. Okay, so she could have switched, but maybe it's still the fair coin. And then a heads. And then a long series of tails. So either she used the fair coin, and she just got lucky to throw, what is that, 5 tails in a row, or she switched to the bias coin because there, 90% of the time, the toss is actually a tails. So one guess might be, right? One guess might be that she had, the first 3 throws, she used the fair coin. Then she switched to the bias coin. Then when she gets some more heads downs here, she switched back to the fair coin. Then she switched back to the bias coin, finally returning to the fair coin. Okay? That's maybe a guess, right? But we don't know. So here, the emissions are the heads, tails, right? And the states are fair, biased coins. So it's like this, right? Here's the hidden Markov model that underlies what the dealer was doing. So here, B means that she's using the biased coin, and fair means that she's using the fair coin. So here, the emission probabilities in the fair coins is 50-50 for heads, tails, and here it's 90% for tails, biased. Now, she may have also switched between coins probabilistically. We don't know that, right? But we don't know her probabilities, but she might have continued to use the same coin with 0.7%, 70%, 0.7, and she might have continued to use the fair coin, 0.8, but she'll switch the coins with probability 0.3 and probability 0.2. And the initial probabilities, oops, this is supposed to be, it's a typo here, this should be 1/2. Okay, so which coin she starts with is equifrequent. So for example, to return to that previous example, okay, for example, this is the previous sequence that we looked at, heads, tails, 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 etc. The hidden sequence here was fair, 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 biased, then fair, then biased, and then fair. My previous guess was fair, biased, it agrees, but then I'm incorrect there. Then I'm correct. What's happened here? They've got out of whack. That's false. I get that wrong. Whoops, what's happened here? Something when I reformatted. Oh, I see. Let me just shift this over, guys. Yeah, okay. So I'm correct sometimes, right? I was correct up to here, and then I get this wrong, that, and that. So I made some mistakes, right? But that would be one valid guess. That wasn't actually the set of coin choices that the dealer actually used. Okay, so if you try, so hopefully you understand that game. So again, the hidden part here is actually what we're trying to guess, right? That's the game. We don't know which coin she was using. All we see is the results dictated by this model of heads and tails, okay? So if you're not sure at this point, or something's confusing you, I'd stop the video and just think about this a little bit, right? Okay. So here's some challenges. Same model. Now, I ask you, can you provide two distinct paths through the hidden Markov model that can produce star? So I wrote star up here for your benefit. And that's easy, right? Actually, any sequence is possible between these two states. So for example, I could have started in the biased. I could have just stayed biased the whole time, right? That's perfectly legitimate. I can still toss coins and get heads and tails, but three heads in a row with the biased coin is pretty unlikely event, right? But it is a valid path through that hidden Markov model, right? So this is all just staying in the biased coin the whole way. Here's another example. I could just stay with the fair coin the whole time, right? Now, what would be more likely, actually? That's a good question. So which of these two possibilities is more likely given this particular star, right? And I would guess that because we have so many heads, 1, 2, 3, 4, 5, 6, and heads is so low probability over here, that it's more likely that this fair coin the whole way is more likely than that she'd ever used than she just used the bias coin, right? Hope you're with me there. Of course, there's a lot of different possibilities of switching to and from the fair and biased. And the second challenge is how many such paths through the HMM can produce star. Okay, so I'm gonna let you think about that one, right? How many different ways, and we'll come back near the end of the lecture with a more kind of example, a more, I'll give you an exact answer to it a little bit later, but you should stop and think about it now. Okay, so here's another challenge. So we can ask the question, what is the probability of a observed set of heads and tails, knowing what the states were? So if the dealer tells you, okay, that she was using the bias coin for the first two tosses and then the fair coin for the second two, and you observe publicly heads, tails, tails, heads, we can calculate the probability quite easily from that. So if we call this X, it's made up of two components, observed and hidden, then the probability of X is the probability that you started in state with the bias coin, okay? Because she started in the state of the bias coin. And that probability is 0.5, right? The same typo is there, right? So, yeah. Now, I started in the bias coin, or she did, and then she admitted a heads. And so what is the probability of admitting a heads using the bias coin? That's 0.1, right? So the emission of H with the B coin is 0.1. Then this says that she stayed in state B, right? So it's the probability of going from B to B, which is this probability here, 0.7. And she has a B, and now she emits a tail, again in state B, so that's 0.9, right? That's this guy. And then we see here that she switched to the fair coin. So she traveled along this arc with probability 0.3, right? That's from probability from B to F is 0.3. Now she's in state F, and she admits a T, and that occurs with probability 0.5. That's that 0.5. She stays in state F. That occurs with probability 0.8. And finally, she admits a heads, and that occurs with probability 0.5. So this, if I was to do the arithmetic, is the probability of this configuration. Okay? So here's a challenge for you. for the observed coin tosses of heads, tails, tails, heads, what is the most what sequence state what sequence of states induces the highest probability? Okay, so now any heads here, right? As a hint here, any heads here is very rare in this model, and over here it's 0.5. But of course, if I switch from, so if I start off in heads, let's do this, let's try this. I start off, heads is pretty likely, so I'm going to say I started off with the fair coin, okay? And so, well, that pi sub A is equal to pi sub B, so that doesn't matter. These guys are equal. And then I emitted a 0.5, right? So we'll get rid of that anyway. So now this is 0.5. Now this 0.5 is better than 0.1, right? Okay. And now at tails, if I stayed in the fair coin, would be 0.5. If I switch to, now it's 0.5 times 0.8, right? So I have two choices. Either I stay in F, and that's going to be 0.8, right, for that self transition, times another head, which is another 0.5. If I go over and I go over to B state, that scenario is a 0.2 times then, sorry, what am I saying here? No, sorry, that was the 0.5 was from before. That 0.5 is there, but this 0.8, if I take the transition instead of staying in the same state, it's 0.2, it's much less than 0.8. But now, the probability of a tails over here, this tails in the bias coin now would be 0.9, so I would pick it up. But however, 0.2 is much less than 0.8, and 0.9 is compared to 0.9 and 0.5. So I would bet that the more probable scenario is that I stay in with the fair coin here. Now I'm still over here, so again, either I transit over to the biased and I get 0.9, so 0.2 times 0.9, or I stay where I am and I take a 0.8 times 0.5, right? So now the thing is that because there's two tails in a row, maybe that actually now makes it worthwhile to just transit once with 0.2 and then you get a 0.7. I'm not sure, but let's keep it like this for now. And so that would be basically me saying I stay in the fair coin the whole way. Is that going to be more probable than the sequence up here, which is BBFF? And is it the most probable? I'll leave it to you to play around, because I think it's a good challenge for you to really make sure that you understand how a hidden Markov model works. And the main thing I want you to focus on here is that there's many different paths through this model that can produce HTTH, but they're all going to have, well, they're going to induce different probabilities, and I want the one that really maximizes it. So that's a really good exercise for you, for your understanding. Okay, so back to gene finding. Essentially what we're going to do is walk along this chromosome, and we're going to, at each step, emit either N for non-coding or C for coding. Okay, and that's going to be done by a hidden Markov model. So at each step, as we walk along the chromosome, we can think of emitting a nucleotide. So an A, C, G, or T as we walk along. And the same thing when we're in the coding region, we emit a nucleotide, A, C, G, or T. Now, what would those emission probabilities be? Would they be the same or not the same between non-coding and coding DNA? And the hidden Markov model approach essentially is predicated upon the belief that the nucleotide composition of non-coding regions is different than the nucleotide composition of coding regions. Okay, so well, before we had heads and tails in our example, now we have a four-way outcome, not just heads and tails, but any nucleotide for both coding and non-coding. But that's not a problem. So how would we set those parameters now? We have a bunch of parameters to set in our model, right? How would we set the emission probabilities of A, C, G, or T in the non-coding region? Well, one way to do it would be to look at our regions that we know are non-coding, okay? And so we would take this region, and this region. This might be from our example set of known, of closely related organism, or from regions of the genome that's been well studied. And we would calculate simply the frequency in non-coding regions of A, likewise for C, G, and T, okay? And then the second obvious issue is, what do you do about coding frequencies, right? And that proceeds analogously. So we would just calculate the frequency of A within all the known coding regions that we have. Okay, so that gives us a frequency for the non-coding and the coding. And again, this whole hidden Markov model approach is predicated on the belief that these two frequency vectors will be sufficiently different that we can predict when we're encoding and non-coding. So let's just stop there for a second and go back to our coin toss problem. You know, if one coin was fair, 50-50 heads tails, but the other coin was just slightly biased, like 0.500000001 versus 0.4999999, right? You know, well, it is biased, so you'd have to probably flip the coin an awful long time to watch. You'd have to watch the game a long time to detect such a small difference between fair and biased. Another possibility, of course, is that the fair coin is 50-50. This is the other extreme, but the biased coin is 99% heads and 1% tail. You wouldn't have to watch that game very long before you kind of know what coin the dealer's using. So we're a bit at the mercy here that these two frequency vectors are quite different. And in assignment 4, you're going to look at the yeast genome and determine that yourself. Okay, so we still have some other parameters, of course. We need to start thinking about these transition arcs, right? That go from non-coding to itself, coding to itself, etc. So what is the probability of non-coding to itself? Let's start there. And, we could look through how often does that occur, and it occurs there and there and there and there, right? It occurs basically 1 minus the length of every run of non-coding regions, right? So that then is really simple to calculate. That's just going to be the number of times that we observe a non-coding and a non-coding side by side across the whole length of this chromosome. And likewise, if we want to calculate the probability of non-coding to coding, we would ask, how many times do we see this? So a transition from non-coding to coding, right? Now we're inside of a gene there, and we count how many thereof divided by the total length of the chromosome, right? Similarly for C to C and C to NC. So C to C would be any examples where we have 2 C's in a row, right? Divided by the total length of the chromosome. And C to N, or any of these examples here, it's basically the number of genes, right? Divided by the total number of nucleotides in the chromosome. Okay, so that actually parameterizes our model. Now we have one last little thing here. We're missing one element of a hidden Markov model, and that's the initial probabilities. And for that, in this kind of scenario here, when we start down here, does it really matter if we start with an A, C, G, or T? Absolutely not. Most likely it's an A or T because the telomeres are highly repetitive regions and they often begin with TAs. But it's meaningless in this model. So we could set pi sub A, C, and G and T all to be the same at 0.25. So those initial probabilities just don't play any kind of interesting role here. Now, of course, we've set that model up, and we have now, let's say, our genome, right, that we don't know. We don't know this part here, right? Now we come in and we have another genome, or a different chromosome, or a different stretch of region that we haven't studied yet, and we don't have that and in C, we don't know whether coding and non-coding regions are. That's what we want to determine. So how do we then use the hidden Markov model to find when we're in the state of coding and when we're in the state of non-coding? And well, it would be very difficult to use the same kind of ad hoc algorithms we used when we were looking at this betting game. A more formal, it's a beautiful algorithm called Viterbi, solves this optimally in a very quick way. Now, the algorithm itself is beyond the scope of this course. It would take a good couple lectures to explain the probability calculations. But we can still understand the intuition of it pretty easily. So our input is going to be our HMM. Okay, I'm going to show the Viterbi algorithm is quite general. It can be used in lots of contexts for any hidden Markov model, but I'll explain it in the context of gene finding here. Our input is our hidden Markov model that we just looked at, and an observed sequence. That's the region that we're interested in characterizing. Okay, just close that. And what's going to output is the most likely set of transitions between coding and non-coding regions. So it's going to basically assign N and C's as we walk along, but it's going to do so in a way that maximizes the probability of that occurring. Okay, I'll explain this in a little bit. This is a very difficult computational problem, extremely difficult. So we talked a little bit about this before, about how many different ways you could have paths, right? And different paths through this Markov model. So if my input sequence is this Markov model, and if my input is this sequence here and my Markov model, how many different ways could I go through my Markov model hidden Markov model to generate A, T, C, and G? So I could be in the non-coding and emit an A, and I could stay in non-coding and emit a T. I could emit a C in non-coding. I could stay in non-coding the whole way, or I could stay in non-coding until I hit a C in the last step for the G. So non, and I switch and I hit and I emit the G. I could have non-coding non-coding coding, and if you recognize this, and you can ask how many on a sequence of length 4, how many possible different transitions are there? It would be 2 to the 4 possibilities, and that's really easy to see if you don't. If you think about it, if you replace n by 1 and c by a 0, these are binary vectors, and so n by a 0. Let's do n by 0 and n by a 1, c by a 1. Okay, and so this first vector is 0, 0, 0, 0. Then we go to 0, 0, 0, 1. And if you count in binary, maybe some of you have done this before, maybe some of you have never counted in binary. It's kind of fun. That's 2, 3, and then 4 looks like this, all the way down to 1, 1, 1, 1, which is the number 15. Okay, so this is the number 15, but we start at 0, and this is 1, this is 2, 3, 4, all the way to 15. So that's 2 to the 4, 16 possibilities, right? Now, how long is a chromosome? It might be 1 million base pairs long. So 2 to the 1 million is an enormous number. It exceeds the number of molecules in the universe, which is estimated to be around 10 to the 80th only. So that's not something we can do. We just couldn't enumerate every possible transition through this system, calculate the probability of that, and then choose the one from this list of 16 that maximizes the probability. And for each such possibility, we would just compute the probability as we did last class, which would be the probability of starting in state C, which is a quarter because it's equifrequent, then the probability of emitting an A in state C, and then the probability of going from C to N, right, taking this transition, and then the probability of emitting a T in state N, and then transiting back to state C, emitting a C, right? And then actually going back to state N and emitting a G. So just like we did last class, you would just chain all those probabilities together. Now, I would think that number 11 is a very low probability event. All those transitions back and forth between non-coding and coding have very low probability, right? Compared to staying in non-coding or coding regions. But one of these 16 possibilities will have the highest probability, and that's the one that you would choose. And from that, you get, of course, where your prediction of where genes and non-coding regions sit. Okay, so a couple concepts here for machine learning, because we're gonna see more and more machine learning now as the course goes on, and this starts here with the idea of a positive and negative learning set. So we know that for this whole system to work, we need some of the genes, either in our target organism, we're trying to find the rest of them, or in a closely related organism. And those examples of both coding and non-coding regions, those examples of the coding regions are what we term the positive learning set. So they're positive examples. And those regions that we know are non-coding, we term the negative learning set. So our learning set, some people might call it a gold standard, but our learning set consists of two sides, positive examples and negative examples. And you should maybe stop the video and ask how that information about positive and negative is incorporated into the hidden Markov model. Of course, the answer is that we form two emission vectors, right? One emission vector in the coding regions and one emission vector in the non-coding regions. And if those two vectors are different, meaning the nucleotide frequency is different between coding and non-coding, then the hidden Markov model should be able to find an optimal path and identify where the genes are. And well, in general, what usually happens is that we start off with some genes that we know. We start off with some predicted genes that we might validate back in the lab or through other methods, and then we'll add those validated genes to our learning set, and then we'll reapply the hidden Markov model again to the chromosome and predict more genes, validate, add those to the learning set, either positive or negative as we go around, and we would keep cycling through this procedure until we're happy that we found all of the genes. Okay, so let's now that's really hidden Markov models sit underneath a lot of different computational biology methods, and certainly for gene finding, but industrial strength software uses other things besides frequency vectors, etc. So we're going to look at some of those things now. All right, the other thing I wanted to talk about today was gene finding approaches and software that go beyond the hidden Markov model that we've looked at. So, well, firstly, there's a division between eukaryotic and prokaryotic, and we'll talk a little bit about why, but it's generally speaking, gene finding is much different in those two broad categories. So eukaryotic with cells, these are cells with the nucleus, and prokaryotic don't. The second, well, I can show you here some of the gene finding software that's out here. This is from Wikipedia. It's not a bad list, actually. You'll see if you go through this that firstly, you'll notice that roughly speaking, you're either a eukaryotic or a prokaryotic gene finder. You're not both. There's some exceptions, GeneMark, for example. If you go through here, you'll see there's a lot that are, in fact, based on hidden Markov models, BGF. Some of these other ones are too, but FGenish. Where else are we here? Come down here to, for example, I've lost it now, I'm sorry. Some of these are also HMM based, certainly Veil, okay? And a number of other techniques are also prevalent here. Neural net based methods. So we're going to look at some neural nets near the end of the course. decision trees, support vector machines, or short for SVMs, etc. So there are different techniques out there. A lot of these are machine learning techniques, though, which means that you fundamentally you have a learning set of positive genes and negatives, non-coding areas, and you use that to train a model. It's just that the model changes. Instead of a hidden Markov model, you use something else. Okay. So yeah, and then there's a broad difference between intrinsic and extrinsic approaches. Now, we've seen an intrinsic approach already. HMMs are one type. And I should say that they're not just for protein encoding genes. You could use them to, say, detect long non-encoding RNAs. Again, it just comes down to your learning set. You're going to reparameterize your hidden Markov model. The transition probabilities between states and the emission probabilities would have to be readjusted for whatever domain you're working in. But of course, you need the learning set of you need examples of long non-coding RNAs versus non-coding RNAs. Well, that's not quite. I don't need to say non as in actual coding RNAs, but at least you need to. You need to know regions of the genome that aren't non-coding RNAs, and it's all based on essentially machine learning. You have that set of positive and negative examples, and you train your model. Now, I mean, fundamentally, eukaryote are more complex than prokaryote, and in a way, then there's more hints in the DNA in eukaryote for where a gene might be, but it's challenging because the genomes are so big. And for example, in vertebrate genomes, you're only looking at 2% of the genome coding for a gene. So there's 98% of the genome is just random or blank. That's debatable, but people call it junk DNA. I'm not sure it's 98%, but it's a large fraction. And so you think about in a bacteria, your promoter is really tightly constrained around the transcription start site. And so all those short binding sites, for example, and they would have sequence properties, right, that you might be able to train an HMM to find. Those things might be compressed in bacteria and therefore easier to find. But in eukaryote, these might stretch thousands of base pairs upstream of the gene, and enhancers and these kinds of things might be even 100,000 base pairs, like huge tracks upstream of the gene. So that means those little signals are going to be spread over a much larger area, and statistically that'll be harder to identify. But eukaryote, for example, have intron-exon structure. Okay, so I've blown it up down here, and you have splice sites, donor and acceptor sites on the that basically indicate where the splice will occur. You have alternative transcripts, alternative splicing forming different transcripts, etc. So, in fact, these kinds of properties, like the start codon, stop codons for the exons, and these 5' and 3' splice sites, you can use either HMMs or position weight matrices to characterize. So these models for eukaryote become, in some sense, hierarchical, that you have really an ensemble of HMMs or classifiers in general trying to pick up on different components of what it means to be a gene in eukaryote. Whereas bacteria, it's a lot simpler. Even the region upstream of genes is a bit simpler, and you have operon structure, etc. So, and the other major thing is that the genomes are relatively small, and there's very little energetic regions, not to mention no introns. And so, it's not really very hard in a way in prokaryotes to find generally where the gene is. You just want to sort of localize exactly where it starts and stops. And that's, of course, not the case in eukaryotes like human, for example, vertebrates, the higher order eukaryotes. This concept of an open reading frame comes up a lot. Genes are most simply identified by the presence of long open reading frames, and that just means that it's really a simple definition where it's just saying, either I see a start codon in bacteria, it'd be like ATG, GTG, TGG, etc., and a stop codon that that should be TAA, THE, or TGA. For example, here in this DNA logo, these are the start codons in E. coli. So you can see that the second and third positions are TG, but A, G, and T are all present in the first position. So there's a bit of a degeneracy between eukaryotes and prokaryotes in that way. That's a very short motif, right? Now, let's go back to, we had this talk in class, in the discussion at least, about being at the Montreal, Bell Center, and everybody guessing, what was it, a nucleotide sequence of like 6 or 7, I think it was. And, already I think it was expected that about every 2 games somebody would win that game, right? But here, it's only 3 nucleotides long, so it's 4 times 4, 16 times 4, 64 possibilities. So basically, all things being equal, you might expect to see an ATG every 64 base pairs as you walk along a bacterial genome. And, well, that's bad news in a way, because I'm not sure it's bad news, but just by random, if you have a genome that's, let's say, 1 million base pairs long, 1 million by 64, that's what, about 10,000 occurrences, I guess. Slightly more than that, I guess. Maybe closer to 20,000 occurrences of ATG just by random chance alone. Okay. So I think Glimmer, that was the first one that I knew about. It's one of the most famous that I know for prokaryotes. It's based on Hidden Markov models. It's supported by the NCBI. So if you were to go out there and sequence a bacteria, or prokaryote in general, you can go to the NCBI, which we all know about, we're all familiar with, and you can copy or paste your sequence, your FASTA file right there into the Glimmer window, and a little bit later you'll get back this kind of response that says, the first open reading frame is from bases 59 to 583, and it'll give you back everywhere it thinks that there is a gene in that genome. Okay, I mentioned previously that there's this difference between intrinsic and extrinsic approaches, and really there's a third technique that's more popular these days based on next generation sequencing, expression approaches. All 3 can be used in both prokaryotic and eukaryotic settings, really, so all 3 really. I shouldn't have said both. Okay, so I don't like these terms, right? I don't think that they're very good, and I kind of think maybe I created them somehow for this lecture, but regardless, let me try to explain what I mean here. Hidden Markov models are an intrinsic method because they take known examples of genes and non-coding regions and they summarize them. In our example in class, we just summarized them basically by their nucleotide frequencies, right? It's just basically saying what's the frequency of A in a non-coding region, and is it different than a frequency of A in a coding region, right? So it does actually use information from outside sources. It's not completely intrinsic, but it does bring in information from outside, but it doesn't bring the whole gene. It's just a summary of the gene. Extrinsic methods, on the other hand, are things like sequence alignment that we're going to start to look at for the next 2 lectures, where you take a known gene, for example, its nucleic acid sequence, and you align it against your target genome where you want to find genes. So the idea is that, if you were to use something like a ribosomal subunit, most likely, I guess it would be just about impossible for the target organism not to have a ribosomal subunit. So what you're doing, it won't be exactly the same, but it'll be highly similar. And so this notion of alignment in computational biology is extremely important. One example is BLAST-X, which is a part of the BLAST family, and it basically will take your gene that you're looking for and try to align it across the entire genome and find where, if anywhere, it aligns well. So these are really kind of complementary approaches. So extrinsic methods are really powerful for finding genes that we already know about, right? So we all expect that we're going to have ribosomal subunits. We're all going to have a proteasome. Well, I think it's probably, some of these things are ubiquitous. Not everybody's going to have a nucleus, but if you're in the eukaryotes, everybody's going to have a nucleus by definition. So you're going to have things like membrane transport, and you're going to have chaperones, etc. So a lot of genes are ubiquitous across the tree of life, or nearly ubiquitous, right? So you can use our known examples of those genes to look for similar ones in the organisms. That's really powerful, but of course, if you have a really novel organism from the bottom of the ocean that hasn't been studied, from an ecosystem that hasn't been studied very well, where there might be lots of novel organisms, there may be genes in there that look nothing like what we've seen before. And those ones won't get picked up by these methods, right? Well, there might be domains that they share, but sometimes they'll be too faint. There'll be the evolutionary divergence may be too far, etc. And you won't get them through these extrinsic methods. Now, intrinsic methods will find those kinds of, in principle, could find those rare, interesting variants, but it doesn't have a lot to go on. It's going on just this metastructure of a gene, like how is the gene started, how is it stopped, what sort of structure does it have in between in terms of introns and exons, is there a promoter, etc. Can we detect binding sites? All those pieces of information go towards predicting whether a locus on a genome is really a gene. So it has the chance to identify novel genes as long as they carry that sort of that intrinsic structure of what it means to be a gene. Well, a third method is RNA sequencing. I won't go into this, but it's pretty intuitive, I think. If you sequence the transcriptome of a cell, of a population of cells, well, what you're doing is you're measuring, most likely, it's the sequencer has actually sequenced cDNA that's derived from the mRNA, and that mRNA is derived from the genomic DNA of the organism, right, from those cells. So by aligning your transcripts that you've sequenced back to the genome in a smart way, you can find those parts of the genome that are expressed. And that really has has benefits above and beyond both the intrinsic and extrinsic methods, because it's really almost less of a prediction, right? It's really kind of direct evidence that that part of the genome is expressed. Now, there's also sorts of complicating caveats and special situations there, but it's turning out to be a very powerful method. It makes sense, right? Well, really, when you go to industrial strength gene finders that are supported by the NCBI and JGI for microbes, etc., the Broad, places that are doing a lot of sequencing, you'll find ensembles of techniques pushed together and mixtures of intrinsic and extrinsic methods. This is, for example, how you might do it in eukaryotes, where you would use homology search from databases like the NCBI RefSec to align against the genome to get an idea where things are. And then you would use bioinformatic HMM-based predictors to predict exon, intron structures, compare them with RNA-seq to look at the transcripts, etc., and cycle through here until you somehow stabilize on what you think are the true genes in the genome, right? And that's these are some of the most famous, most powerful ones currently for doing that. HMMs and R, well, really, I don't have experience with this DepMix S4. I think it's pretty famous. Of course, hidden Markov models get used in domains outside of biology. I think Seek HMM is for social networking or something, like not like Facebook, but social data. I'm not entirely sure. I've never used those two techniques. I'm sure they're very good packages. I know more Hummer, which is the bioinformatics computational biology software. There is a R package for it, so I think that it's pretty easy to set up hidden Markov models really quickly. And I mean, right now you guys have all the capacity to do that yourself. You have all the components. You would just need to look at the manual and try to massage your data into one of those frameworks. Okay, so points of reflection to end here. Think a little bit about what the relative advantages and disadvantages of these different methods are. Include when you're in your thinking about RNA-seq and how that could be used. Make sure you understand the basic idea there. If you don't, we can talk about it in the lecture. Okay, so next class we're going to start to look at, we're going to carry forward with the Markov models, but towards this sort of in line with these extrinsic approaches of sequence alignment and modeling evolution, particularly in cancer. I'll see you then.